but other features like C-peptides, triglyceride, the diet score, and the response to glucose at breakfast. And then we found, we started surveying these to all the data we had available in the lab, and Elisa in the lab took up on these tasks. So we looked at prevalence, worldwide prevalence of Blastocystis. We found some specific patterns that were already suggested by previous work, but in this case, we were able to analyze more than 56,000 human gut genomes. We found association with different dietary patterns, but more interesting, in meta-analysis settings, we also confirmed the association with BMI. So people with higher BMI tend to have less chances of having Blastocystis in their gut microbiome. And again, at the end, we like to use machine learning. So what we were trying to do is, can we predict Blastocystis presence or absence based on the microbiome composition, only looking at the bacteria and archaea fraction? And we saw that when Blastocystis is present, we usually have species here that are more favorably linked with cardiometabolic health, and when Blastocystis is absent up here, we have species that are unfavorably ranked. The last part on FMT has been talked quite a lot already today. So what we did here just a couple of years ago was to try to define a strain sharing pattern looking at the metagenomes between donors and recipients. And so in this case, we collected and reanalyzed a lot of FMT plus a couple of data that we specifically sequenced for this project. We defined a strain sharing rate between donors and recipients, and we were linking, for example, this engraftment rate of strains to a better clinical success. So if the recipient had higher strain engraftment, that was linked with better response to the therapy. But what we did again using machine learning was to try to predict, for example, alpha diversity. So in this case, we can predict alpha diversity in the donor and in the post-FMT just by using the microbiome composition of the two different individuals. And what you see here is also how we are able to predict how well the engraftment will be given the microbiome composition of the donors in that specific recipient. So I hope I convinced you that we need much deeper profiling and characterization of the microbiome members. Machine learning, it can be very useful. We need to be careful in applying it, but it can provide us a very nice perspective and a different point of view on microbiome association. And again, having now the